Good afternoon, everyone. It's a great honor for me to welcome you to this session dedicated to the advancement in basic and translational research in Angelman syndrome. We have 5 talks scheduled, each lasting 30 minutes, plus a 10-minute discussion. I would like to thank in advance all the speaker participants for their contribution. So, let's begin. It's my pleasure to introduce our first speaker, Professor Cristina Alberini. She is a leading figure in science of Angelman syndrome, and she is a full professor in neural science at the New York University. She has been professor in New York University since 2011. Professor Alberini is also an elected member of the American Academy of Arts and Sciences. Professor Alberini research focuses on the molecular and cellular mechanisms underlying long-term memory formation and their role in cognitive impairment, neurodevelopmental disorders such as Angelman syndrome, and neurodegenerative diseases. The title of her talk is From Bench to Bedside, Targeting IGF-2-CIM6P Receptor to Develop Novel Treatments for Angelman Syndrome and Other Neurodevelopmental Disorders. Cristina, the floor is yours. Thank you.
Thank you so much, Monica, for the wonderful introduction. I'm very happy to be here with you today. I want to thank Benedetta and all the organizers of this conference for such a wonderful job. And I'm very delighted to share with you a project that has been going on in the lab for a number of years now that started from the discovery of molecular mechanisms of long-term memory and long-term plasticity. So the lab has been working on the biology of memory for more than 30 years. I really started at the beginning of these questions. And the memories that we are working on are important for cognitive functions. These are the memories that are processed by the so-called hippocampus-dependent memory system or medial temporal lobe-dependent memory system. These memories are the memories of who, what, where, and when. Who did I meet today? What did I do? What did I see? Where was I? And binding all these elements together so that we have a narrative of our experience. And these memories are recalled consciously. So when I talk about memory, you immediately think about this type of memories, the memories of the events of your life. So that's what we are doing in the lab. And these memories, as I said, are key for cognitive functions. If we don't develop these memories, we have no ability to have cognitive functions, including decision-making. If you think about decision-making is the process that relies on our history, all our experiences. So enough of memory, almost. Rodents, like humans, use the same part of the brains to process this information, meaning the hippocampus, cortical regions, and a number of other regions. So we go into the brain of rodents and try to find out what changes in these regions with learning and what is required for long-term memory. Particularly, we studied the hippocampus for many, many years. Now we are moving to other areas. Okay, so in the number of these studies, we found, and this was an unbiased approach, meaning we didn't have the hypothesis that this gene was important, this molecule was important. We found that a molecule that is a small protein, is called insulin-like growth factor 2, or IGF-2, is upregulated. There is more of it in the hippocampus after learning. So if you learn something new today, which you are, you're going to increase the expression of IGF-2 in your hippocampus. If we block that expression, long-term memory was completely blocked. So IGF-2 turned out to be an essential protein for long-term memory formation, but not only. When we supplied IGF-2 with learning or memory retrieval, we produced a spectacular memory enhancement, a significant memory enhancement. So IGF-2 is not only required for memory, but is also a very strong memory enhancer. So I'm not going to go into the data of learning and memory and IGF-2 because we have been studying this for more than 20 years, and we have a lot of studies on it. But sufficient to say, to continue in our talk, that IGF-2 luckily crosses the blood-brain barrier. We were talking about this morning, because there are transporters in the blood-brain barrier. Binds IGF-2, takes it into the brain. So we can inject IGF-2 in rodents, and these are mice in these experiments. We inject IGF-2 subcutaneously, and we look at the effect. And as you can see here, in blue is the control groups that received vehicle injection. And in the pink groups, you see the effect of IGF-2 in different types of memories, mice. Contextual fear conditioning, novel object recognition, social recognition memory. You have heard about these, a couple of these tasks, the contextual fear conditioning and the novel object recognition in the morning. Social, it means remembering somebody conspecific. Okay, so all these memories, as I said, is about who, what, where, and when. And all these memories are significantly enhanced if I inject IGF-2 before or after training or before retrieval. So what is this great memory enhancer? What is IGF-2? IGF-2 belongs to a system known as IGF insulin system. And together with IGF-2, we have two other proteins, IGF-1, insulin agrofactor 1, and insulin itself. They bind to receptors whose affinity takes the name of the factor. In other words, we have three types of receptors. We have more of that because they can assemble with each other. But let's say for simplicity, three types of receptors. The insulin receptors, IGF-1 receptor, IGF-2 receptors. Insulin receptors and IGF-1 receptors, which binds with high affinity respectively to insulin or IGF-1, are the classic receptors for growth. So they stimulate the signal transductions that leads to growth. IGF-2 receptor is very different. IGF-2 receptor doesn't have a known signal transduction. It's a single transmembrane receptor, high affinity for IGF-2, low affinity for IGF-1 and insulin. And this receptor takes IGF-2 inside the cell and takes it to lysosomes, the stations that degrades the protein, that dissolve the proteins. So it's a receptor that takes protein to lysosomes for degrading them. It has more functions than that. We're going to see this in a minute. But this is the classical, what's classically known about IGF-2 and IGF-2 receptor. So it turns out that after our study identified IGF-2 as an important protein for learning and memory, a number of studies looked at diseases. Because if IGF-2 is a memory enhancer, can we actually use IGF-2 in cognitive impairments, in problems of memory, to increase the memory and cognition? So there is a large literature showing that IGF-2 has beneficial effect on aging. I'm going to show you one set of data. Neurodevelopmental disorders, including Angelman syndrome, but also neurodegenerative diseases, Huntington, Alzheimer, and also other type of diseases like schizophrenia, stroke, repair from stress, depression, motor neuron degeneration, and ALS. I'm happy to send you the references. There is a very long list of publications on all these diseases. So I want to also remind that IGF-2 is highly expressed during fetal development, and then the level reduces in all tissue, most of the tissues, but remains relatively highly expressed in some of them. One is the brain. So it's unclear what does IGF-2 do in the mature brain. And one of the functions we found is required for long-term memory formation. Okay, so these are 3 examples from our lab, but as I said, there are many labs that have worked on it, showing the effect of IGF-2 in a mouse model of autism spectrum disorder. The model is called BTBR, and is a model that has the phenotypes, the behavioral phenotypes of classical autism. So they have, for example, social impairments, cognitive impairments, repetitive behavior, seizures. We studied that model and injected subcutaneously, subcutaneous injection, IGF-2. And you see this in the graphs, that the injection was done 20 minutes before training, before a learning event, and in this case it's contextual fear conditioning. And 24 hours later, we are measuring the memory, which is measured by freezing. So the higher the level of the bar, the more is the memory. So in the wild type, normal mice, you can see the level of the memory injected with vehicle, and you see that IGF-2 significantly enhance memory. I already showed you that. The BTBR mice have an impairment compared to the normal mice in this memory, and IGF-2 reverses the impairment. So we then moved to Angelman syndrome because we wanted to have a genetic model. And we repeated the experiments. And in this case, I show you another type of learning task, which is object recognition memory. So they basically learn to discriminate a novel object from something that they have already seen. So in this case, you measure the preference. And again, the highest is the preference, the better is the memory. So as you can see here, the vehicle injected wild-type mice has a certain level of memory after learning, which doesn't change with IGF-2 at this time point. It changes if I test other time points. The Angelman mice have an impaired memory, and IGF-2 reverses this impairment. The third example is aging. We took aged rats and injected IGF-2 directly in the hippocampi, in this case, and compared them to young rats. And as you can see here, the aged rats have memory impairment relative to the young, and IGF-2 enhances memories in the young animals and reverse age-related memory impairment. So a number of studies in many different forms of learning and memory, even aging here, shows that the effect of IGF-2 is very potent. When we tested what is the mechanism, what is the receptor to begin with, through which we see these effects, it turns out to be IGF-2 receptor, not the other receptors that have low affinity, not IGF-1 receptor. So IGF-2 receptor is the target here, right? But I show you an effect on so many diseases. What is this, a miracle cure? We were the first to ask the question. We need to understand what's going on in order to understand what we are looking at. And as I mentioned, many diseases now have shown to have a beneficial effect with IGF-2. These are 2 groups, neurodevelopmental disorders and neurodegenerative diseases, but in addition also traumatic brain injury. So what do they have in common is actually protein overaccumulation in the brain. All these diseases have an excess of proteins that accumulate in the brain. So now we start to make sense. Perhaps this mechanism has something to do with protein metabolism. Protein metabolism disruption is very well known to be a marker, a biomarker, a molecular problem in many diseases. A large number of papers have documented this in neurodegenerative diseases. Now, finally, we're understanding the neurodegenerative diseases is not the accumulation of that particular protein. Is not amyloid, is not tau, is not synuclein, is not Huntington. These are different diseases, of course. There is something upstream, and this upstream is acidification of vesicles and autophagy. This has been documented now in many, many studies. Autophagy is the mechanism that when there is an excess of protein or proteins that aggregate or organelles that needs to be eliminated, or something else, by the way, neurons, creates a membrane structure around the cargo, the proteins to be eliminated, takes them through a number of steps, fuses with the lysosomes, and in the lysosomes there is protein degradation. Autophagy is not just that. It turned out in the last several years that actually is required to keep neurons in a good shape and functioning. It's not just eliminating something that is not wanted, but it's a process that with activity, like a learning event, is activated, makes a number of changes which still we don't know the targets of, but this is required for the brain to function physiologically, to function in a good way. Here the picture is showing synapses. And we had talked about synapses in the morning also with Alison. I want to remind here that the brain is not only about synapses. I know that neuroscientists are going there, but please, let's keep in mind that the brain is not just neurons, it's not just synapses. We have a bunch of other cell types that actually needs to be explored. They work all together, by the way. So our target is mannose-6-phosphate receptor, actually cation-independent mannose-6-phosphate receptor, which is the other name of IGF-2 receptor. And this receptor here, there is a schema, has a number of domains. And what we need to know today is that there are two main ligands to this receptor. One is IGF-2, which binds here in domain 11, and the other is mannose-6-phosphate, which binds to other part of the receptor, not the same. And there are at least 3 sites, maybe 4, that binds mannose-6-phosphate in this receptor. This receptor is highly expressed in neurons in the brain. So if we look at the different cell types with markers of different cell types in the brain and IGF-2 receptor expression, what we see is that the majority of the receptor is in neurons. Excitatory and inhibitory neurons is not in microglia, is not in astrocytes. We are now carrying out a complete, as much as possible, survey of where the receptor is expressed in the brain. And if you look at a sagittal, a section that goes from front to back of the brain of the rodent, you see the receptor in all the neurons. They light up like crazy. So there is a lot of receptor in neurons. The receptor is known to have a function. As I mentioned, it binds IGF-2, takes it to lysosomal degradation. But that's not the only function. This receptor accumulates in the trans-Golgi in the cell. It's a subcellular organelle near the nucleus, which is very important for directing the proteins all around the cell, newly synthesized proteins. So this receptor is binding proteins that are newly synthesized and modified with mannose-6-phosphate, takes them around in the cell, takes them to the plasma membrane, takes them to the lysosomes, and all of this trafficking occurs in vesicles. Now, I'm going a little bit here in the details because there is going to be, I hope, an interesting discussion about what's going on. So now, I'm focusing on autophagy, endosomal trafficking, but we're going to hear, and we have heard already, that UBE3A, which is what is deleted in most cases, is actually an enzyme important for another protein degradation system, which is the ubiquitin-dependent, the proteasome. So these are two independent systems. They do, at the end of the day, they do a similar function, which is eliminating proteins that are not needed anymore, or they have to be gone for the system to work. And now it emerges that both of these systems have something going on in these diseases, neurodegeneration and neurodevelopmental disorders. But guess what? Going back to Angelman syndrome, there is a lot coming up on the crosstalk of these systems, even though they have been studied independently until now. Okay, so what's our solution and our explanation is, if we target IGF-2 receptor by either IGF-2 or mannose-6-phosphate, which we used also as a ligand, What we do is that we activate the receptor endocytosis, goes into the cell via vesicles, endosomes, and all this trafficking of vesicles can end up to lysosomes for degradation, can cross-talk to autophagy, again going to protein degradation, but also we have seen that is upstream the regulation of new synthesis, new production of proteins. In other words, this is a master regulator of protein metabolism in neurons. We have published studies showing how autophagy is increased, augmented with learning required for long-term memory in physiological conditions. So in other words, when the brain works, the neurons are making new proteins and eliminating other proteins. There is a balance at the end, but these are specific proteins for whatever activation of those neurons has happened. So the proteins are different in different parts of the brain. It's not the same proteins. Different circuitry is going to regulate different protein metabolism after stimulation, and it's going to regulate the degradation of particular proteins in that circuitry. Okay, so what we did was to use a lot of IGF-2 in experiment, but then go into other ligands like mannose-6-phosphate. We saw the same effects. Mannose-6-phosphate enhances memories. Mannose-6-phosphate reverse problems in a number of disease models, and not only cognitive. If we look at motor problems in Angelman syndrome, we reverse motor problems. If we look at repetitive behavior, we do that. IGF-2 has an effect also on seizures. So there is a global effect, and you can understand why. Here we are targeting some important molecule that regulates quality control of proteins, the distribution, the degradation, and the synthesis, and it's highly enriched in neurons. So we think that in the diseases, you can have a problem in any of these steps, genetic, environmental. If you target one of these steps and you impair them, you're going to end up with protein accumulation in the brain. That's why the protein accumulation in the brain is our common problem here. Okay, just to show you a couple of experiments. When you see red here, is the hippocampus of a rat, which incorporates puromycin is a substrate that tells you that there is protein synthesis. The more red, the higher is protein synthesis. When there is a learning event, trained versus untrained, there is an increase in protein synthesis. That was known. It's very well studied. When we block IGF-2 receptor in the brain, in the hippocampus, that increase is completely blocked. And you see the quantification in the graph downstream. So we then moved on because we want to understand more, but also develop something that can be helpful for new treatments. So we have designed with chemists, collaborators at NYU, Professor Dirk Trauner and Chris Arp, small molecules that derive from mannose-6-phosphate, not IGF-2, mannose-6-phosphate derivative. This is called PMP1. The way that they designed this, PMP1 is a prodrug, which means we can hopefully give the prodrug either via oral administration or subcutaneously, and hopefully it works. And that's the case. I'm going to show you just one example. So PMP1 relative to IGF-2 increases autophagy in the brain. So in this case, we took mice that has a reporter system in the brain that tells us how much autophagy happens. And the system is, just for the science here, is a fusion protein of RFP, red protein, GFP, green protein, and a target to autophagosomes, LC3. In autophagosomes, these two colors, red and green, they make yellow. But when autophagy moves and goes to more acidic vesicles, the GFP is quenched, so you see only red. So by counting yellow versus red vesicles, you have a sense of the autophagic flux going toward the lysosomes. So we did that, and you can see this is some example of the staining in the hippocampus, untrained and trained animals treated with IGF-2 or PMP1. You can see the quantification in a number of brain regions. Both training enhances autophagy, which we know, and treatment enhances significantly autophagy. IGF-2 does it, PMP1 does it, and even better. So in Angelman syndrome, we found that in line with the problem of protein degradation, there is accumulation of proteins in the brain. So here we use the mouse model that was done by Jiang. You're going to hear from him later today, I guess, or tomorrow, I forgot, today. And this mouse model has been used by many, many labs. It's a deletion of UBE3A from the maternal allele. And using this mouse, we have studied a number of behaviors as well as molecular mechanism in the hippocampus. So we're going into the hippocampus of these mice relative to normal, wild type, and looking at certain proteins. These proteins are called immediate early genes and are very important for the plasticity of the brain. 3 of them, ARC, FOS, and EGR1. And what we found when we compare Angelman mice, the hippocampus of Angelman mice, to that of normal, wild type, we found an accumulation of these immediate early genes. ARC accumulation was already described by others before us, but we found that also the other immediate early genes accumulate in the hippocampus of mice in vivo. When we look at the hippocampi after learning, there is a typical increase in immediate early genes in the wild type. This is required for plasticity. There is no movement in the hippocampus of Angelman mice. So in summary, Angelman mice have an overaccumulation of these immediate early genes, and with a learning event, they don't change. The level is kind of stuck. We did a number, actually, of other experiments I'm not going to go into. This is not the only proteins that accumulate in the brain of Angelman syndromes. There are receptors. There is increased level of protein synthesis, increased level of autophagy proteins, no dynamic regulation. Another set of experiments, just to give you a flavor of these studies, is by looking at the reporter for autophagy. As I said, M-cherry, GFP, LC3, you can visualize them because they fluoresce. And you can count the number of yellow versus red puncta in the vesicles. And when you take wild-type mice that either remain in the home cage or undergo a new learning event, you can actually follow up the autophagic flux. And here you can measure the numbers and just focus, for the sake of time, to the panel in blue. There is in the wild-type mouse, there is the ratio of increase of red puncta over yellow, therefore the flux. You see the learning increases significantly autophagic flux. The Angelman mice have a very low level. There is no change in the flux. In other words, we concluded that there is a problem of autophagy. It doesn't move. It doesn't work well. And on the right side, you see accumulation of these structures, which I'm going to call aggregates, even though this is a reporter. They are not endogenous aggregates. But you can see how much in Angelman syndrome and train shows this big chunk of colors. These chunks are bigger amounts of proteins, or actually what they reflect of the lack of movement of autophagic flux. So I'm going to show you a couple of experiments demonstrating how IGF-2 or PMP1 reverse the problems in Angelman mice. This has been an effort of 3 postdocs in the lab, Emmanuel, Janina, and Adam. This is the impairment of contextual fear conditioning in Angelman syndrome. I already showed you the novel object in the slide before. This is the effect of IGF-2. enhancement in normal mice, recovery of the impairment in Angelman. This is the rota rod. You have been introduced to the rotating rod. Of course, if there is a motor coordination problem, they fall off the rod more quickly. And so we measure that in wild type and Angelman. Here is the impairment in the rota rod of Angelman mice. This is the effect of IGF-2, full recovery. We tested this 20 minutes after a subcutaneous injection of IGF-2. We retested 24 hours, 2 days later, and then 4 more days later, the effect is sustained. Okay, so in sum, these are just 2 examples. We did an extensive work with IGF-2 as well as mannose-6-phosphate as well as PMP1 and found that they reverse a number of behavioral responses. IGF-2 and mannose-6-phosphate on spontaneous alternation, marble bearing, tail suspension, clasping, but not on open field. That's the only one we didn't see a change with the treatment. Similar effect with mannose-6-phosphate. Now, we want to develop treatments. Oh, before I go that, I have to mention this. So some of you, I'm sure, in the field know that there has been a paper published a few years ago by Berg et al. Is from the lab of Jill Silverman, showing that insulin-like growth factor 2 does not improve behavioral deficits in mouse and rat models of Angelman syndrome. Although I have shown you that we use many ligands to the receptor, I think I should comment on the IGF-2. So they employed a NURAT at that time, Angelman syndrome model, and a mouse model of Angelman syndrome, which is similar to the one we used. They observed some effect of IGF-2 treatment on EEG, but no change in most behavioral responses, and concluded that IGF-2 does not improve behavioral deficits in rodent model of Angelman syndrome. So, of course, we care a lot about this. We read the paper carefully, and I have to say that you can go and read the paper, and unfortunately there are a number of methodological flaws in the paper, and unfortunately they didn't let me reply to the journal, and so now I have to explain in talks that the study lacks necessary controls and validations. There is a lack of dose response for IGF-2, which is required in a new species, except for the EEG. None of the behavioral tests have run those responses. They actually use different protocols than the one we used in the mouse. This is stated in their discussion. The efficacy of IGF-2 was not tested, which for a negative data is important. And it's unclear why also they did not validate some of the tasks, the effect on the task that other labs, many other labs have shown. They also showed some positive effect on EEG, but they didn't follow up. And the AS rat model, which is great, I think rats, I have been using rats for all my life, are fantastic, but they certainly need more validation. So off of that, I'm gonna show you and conclude the effect of PMP1. So this is not IGF-2, it's mannose-6-phosphate derivative. It's a prodrug, and I'm gonna show you a couple of examples of the effect via oral administration. By oral gavage, if you look at novel object recognition, and you look at wild type injected with vehicle, this is the retention curve on novel object recognition. Angelman mice have problems, they have no memory. Wild type with IGF-2, memory enhancement, prolonged. This is only one single oral gavage, and this is Angelman mice. I know, the data to us too, they are like evoking a number of questions that we need to understand for why we see this prolonged effect. Hind limb clasping, it's another measure of motor problems. So in the mouse, in the rodent models, you can use a very simple test, which is you suspend them by the tail. And if they are normal, they extend the hind limb. If they have motor problems, they clasp them. So you measure clasping scores. And of course, in the wild type injected with vehicle, there is no clasping because they're normal. This is the clasping score of Angelman mice. This is the PMP1 oral gavage in wild type, and this is the effect of PMP1 on Angelman. So you see that they normalize for several weeks, and there is a significant effect up to 4 weeks. It goes back to disease level by 5 weeks. This is a single oral gavage. So this prolonged effect needs, of course, an understanding. Of course, it has to do with plasticity and the mechanism of IGF-2 receptor. So NYU has filed a number of patents. 8 have been issued in the US, in Europe, and other countries. We have a number of pending foreign and US applications, and exclusive license can be given for all indication. So the idea now is to develop these compounds, these small molecules. One is PMP1. We have also backup non-prodrugs. We have run synthesis, which so far so good. We have run in vitro DMPK, also with no alerts, and in vitro safety, also with no alerts. Of course, there is a way to go here, but hopefully with somebody interested and funding, it could be an interesting development for all diseases with protein accumulation in the brain, including Angelman syndrome. So I think I'm out of time. I'm sure I'm out of time. Okay. I want to thank who actually did the work. Here there is a list of gifted, super gifted postdocs and students in the lab. Of course, the funding agencies, the collaborators, and you for listening. Thank you.
Thank you, Cristina, for your excellent presentation. And you took us in the wonderful world of IGF-2 and its receptor. So we have time for questions or comments. Yes. The microphone.
Or just shout.
I was wondering, PMP1 and IGF-2, did you observe those different effects? Do you know that the effects of those different, the effects observed on learning and memory? Thank you.
Yes, yes.
And my second question is, I guess that an excess of autophagy is not a good thing for cells. So do you know if there is a threshold in terms of dose where you observe deleterious effects on cells, for example, excess of apoptosis or neuronal transport defect or metabolism defect?
Very good question. So I'm going to start with the second. We have done also repeated doses for 5 weeks, 3 times a week, either with IGF-2 or mannose-6-phosphate. We're going to do now PMP1. So far, we have not seen any problems. Of course, we need to go inside and look at the details. But I can tell you that with mannose-6-phosphate or IGF-2, which we have run for several weeks, as I said, they seem to even improve other symptoms like GI problems and weight gain. So we are very surprised. I didn't even know that Angelman syndrome had weight gains. The postdoc came to me and he says, they have weight. So, stay tuned. The doses are low. So going to the first question, the doses are low. 30 micrograms per kilogram of IGF-2 sub-Q, so very low. Mannose-6-phosphate, 850 micrograms per kilogram. PMP1, the same. I'm talking about the optimal doses here. We ran dose response, of course. With oral gavage, we can have the same beneficial effect with a lower dose. So because the doses are so low, actually it's going to be difficult to detect them, by the way, in the brain. That could be an explanation for why we don't see aversive effects so far. So fingers crossed.
Thank you.
Question?
So do you know if IGF-2 also passes through the BBB of humans, and does it stimulate autophagy in human neurons?
Yeah, okay. So yes, it does. In humans, they use human BBB, blood-brain barrier, in vitro to look at the transport. They didn't look in humans alive. Does it have an effect in autophagy in humans is not known. We are the first one to figure this out about the effect of IGF-2 in the brain, in brain functions as well as in diseases, what the receptor does, where is it expressed in the brain, and so on. So it's not known yet, but hopefully somebody will do it.
It's just a curiosity. This kind of therapy that potentially ameliorate the metabolism of protein could be potentially available in all kind of genotype in human, I refer.
Yeah, so thank you actually for the question. That reminds me to say, this is not going toward repairing the genetics of Angelman syndrome.
Exactly.
Obviously, right?
It's a
this
quality
is a
therapy.
pharmacology. It's hopefully treating the symptoms. So if the symptoms are due to this overaccumulation, and hopefully these drugs can clear up the overaccumulation and have the same effect we see in mice in humans, well, that's fantastic, right? So ideally, is important for all the forms of Angelman syndrome, as well as other diseases that have accumulation of proteins in the brain.
And the second curiosity is if you are interested to study other pathway. I don't know. I know that there are similar study on GABA signaling, for example. You can give us some information of other potential pathway that could be involved, and if you are interested to study other pathway that potentially are, that potentially can connect different pathology, like you show Angelman, but also other disease.
Autism,
exactly.
Absolutely, yes. So this is another thing that reminds me to say something. Thank you so much. Receptors, like GABA receptors, are actually modulated by plasticity in the sense that there is synthesis and degradation. This receptor is a master regulator of transferring around proteins, including receptor and recycling. It's on recycling endosomes, this receptor. So not only is going to control the newly synthesized receptor, where they go, but also how they recycle. So here we are in a mechanism that is upstream of a number of receptors, not only GABA, a number of them. Yeah.
Thank you.
Okay. So yes, I'm interested in looking at the receptors.
Okay,
Sure,
thank you, Cristina.